This is the final video in the cells as the basis of life topic, and it's concerned with cell culture. So cell culture is all about how we as scientists use the growth of cells in culture, which is separated from its natural environment in order to grow cells to study them, but also use them in various applications. So cell culture is all about getting a bacterial, plant, animal, including insect and all sorts of mammalian cells to grow in cell culture. And so here's some different examples of what cell culture looks like and the different techniques used. This picture here refers to bacterial cell culture. We either grow them in liquid or solid medium. The difference between them is only just that this one has agar, which is a jelly-like substance, but in general what we've got here is some nutrients, amino acids, sugars and things like that that the cells can grow on. Sometimes there's also some other colored things in here which might be indicators or some blood extracts, but this is an example of some bacterial cell cultures growing on petri dishes, which obviously we're very familiar with on a nutrient agar. So the reason for growing bacterial cells might be, for instance, we look at biotechnology. We are transforming bacteria with plasmids and growing them in order to grow up and clone plasmid DNA. But more frequently, obviously, we look at growing bacterial cells in medicine in order to look at samples. So when you have a swab taken to see what type of infection you might have, they can grow bacteria, and depending on how they look and how they grow and other characteristics what they grow on, then scientists, in fact, we're talking about microbiologists, are able to identify what kind of pathogen might be growing in your body and give that information back to your doctor. Here we have some plant cells growing on a petri dish. So when they're growing in this undifferentiated state, they don't really look much like plants, but from these we can then add some hormones to get roots and shoots to start growing and transplant these. So again, in the biotechnology sense, we were talking about growing cells in culture and transforming them by blasting a gene gun into there. With plasmids on it, and then this plant growing out from this might have the gene of interest in it, and you can use those. But of course, also these cells might be growing to study them and understand plant biology more. And similar kind of thing with human cells here. So we tend not to grow them on solid medium. Instead, they grow in a liquid medium but attached to the bottom of the dish. So this is a microscope view of some cells that might be growing on the bottom of this dish. And this culture medium, again, like the liquid medium we have here, is full of nutrients and provides all of the sugars and amino acids and other things that these cells might need to grow. And much like blood, this has to be passed over the cells, so usually they sit in there for a day or two, and then fresh medium is added with more nutrients and the removal of wastes. So the idea of cell culture is very much to reproduce the conditions inside the body in which this might be growing. So you have a warm temperature, provision of nutrients, removal of wastes, and gas exchange with oxygen and carbon dioxide. This is what cell culture techniques look like when the dishes are out of the incubator. And so here there's some dishes with some mammalian cells growing in them. The important thing to remember is when you provide nutrients and warmth and all of those good conditions for instance, human cells or mouse cells to grow, that's the perfect conditions for bacteria to grow as well, and they will grow faster than the mammalian cells. And so it's very important to maintain what's called aseptic technique, which is basically just keeping everything sterile. So around this picture we can see multiple things that relate to aseptic technique. So firstly, we're doing this in a stainless steel cabinet called a laminar flow hood. This can be swabbed down with alcohol to sterilize these surfaces really easily, and in fact that's a UV light there as well, which will irradiate the area and make sure nothing is growing in between uses. When you open it up, you take off the barrier here, but only your hands and the instruments you're using go in. This plate obviously protects you, the air from you breathing out bacteria onto there. And there's also a flow of air that comes down through filtered holes here, and then is sucked into a vacuum here, so that even air floating from the outside will be sucked down here rather than go into the dishes. And that air is filtered to remove all bacteria from it as well. Clearly we have gloves and lab coats here, and a lot of this being used is sterile plasticware that is sterilized before use and then thrown away. Here we have a container that contains some of these glass pipette tips, which may be reused, but they go into these containers to be sterilized at high temperature and pressure in between use as well. So lots of things here to keep this area sterile, and then the lids go back on quickly, and no bacteria can get into these culture dishes, otherwise they will be ruined and have to be thrown out. So it's expensive, it's tricky. So why do we want to grow mammalian cells? So first of all, obviously to study them, but here's an example of the use of mammalian cells in medicine. So if you wanted to develop a new drug, you don't just give drugs to people or animals first, you want to grow them on cells and see how they go. So here's a sort of starting chemical compound of 1500 compounds that are chemically related that you might want to put onto some cells and see how they go. So here's the source cells. You grow those in multi-well dishes. If I just flick back, you'll see this is what's going on here, is that she's putting a small amount of cell culture into these that contain less than a milliliter of cells growing in there. So you can check multiple different batches of cells at once. It's kind of like 96 test tubes all in one little batch. And you could have multiple plates like this. So if 1500 compounds are put onto these cells, we know that some are going to be inhibitors of those cells and others you don't know about. You can then check the viability, how healthy and how well are these cells growing, and then compare to see which ones increase cell growth, which ones decrease cell growth. The aim might be to stop them growing. If these are cancer cells, I think this one was a pancreatic cancer cell, you actually want to stop them growing. So you're trying to find the compounds that are best at reducing the viability and stopping cell growth. Of course, you'd have to go through multiple other tests before you started thinking about using this as a drug, but this is how we can do a screen of many compounds to see what kind of effects they have on cells by just putting them on a small batch of cells grown in culture before giving them to live animals or humans. Other uses of mammalian cell culture would of course be some of those things about biotechnology and gene therapy, tissue regeneration, lots of medical uses, and of course basic scientific research about understanding the processes that happen inside cells so that we can better understand ourselves and improve medicine.